Hi Hires, we are looking at key area 6, A and B, so we're looking at what mutation is and single gene mutations. Okay, so what is a mutation? Mutation is quite simply a change in the DNA of a cell. The National 5 section on this on mutation is a nice treatment of it and is a very nice step up, so if you want to go back and look at that and then move back to this, that's not a bad thing. So from a higher point of view, what you need to recognise is how it actually alters what's happening. So it might change a protein or it can stop it being produced, and those are two different things, and we're going to look at this in quite a bit of detail. What you should reckon about mutations is that they're not targeted, they are random. They will be at any point in the DNA, and they are spontaneous and they are rare. Okay, so there's an example there of 0.003 mutations per genome per cell generation in bacteria. Okay, so that's a rate, but we don't know where it's going to happen, and that's important, especially when we look at molecular clocks a bit later on. So anything which can increase the rate, we should recognise already. You know what a mutagen is. That's any kind of ionizing radiation, so basically anything down from violet, so your ultraviolet, your X-rays, your gamma rays, but you also have chemical mutagens, so these are things that affect the DNA from a chemical point of view. So the first type of mutation that you need to be able to talk about is single gene mutations. So that means that you're looking at a specific point inside the gene code. So we're only looking at one gene, so what we're looking at is the nucleotide sequence. Okay. There are several ways that you have to talk about this. So you have to be able to talk about what's happened. And there are only three things that we're looking at here. You can either do substitution, insertion, or deletion. Or also you can describe the impact that they have. So what actually has happened to the DNA code when you've done one of those things. So we have words like silent, missense, nonsense. We have things like splice site, and we have something called a frame shift. Okay, so these are, sometimes you will use one or more of these words, all to do with describing what's happened with the substitution, insertion, or deletion. Right, so what we're looking at here is literally a change in the base sequence. Substitution, insertion, deletion, I think they make sense, but I'm going to look at each of these in turn. You have to remember those three. It's SID, SID. Here are some famous SIDs, if that helps. So substitution. Substitution is exactly what it sounds like. You swap out a base. So at one point in the sequence, any point in the code, you swap an A for a T, or a T for a G, or a C for a G, or whatever, anything. You've just taken one base out and swapped it. Because DNA coding has some redundancy, doesn't always do what you expect. Like, it might not actually do anything, but a bit more on that later. Okay, so when we're talking about impact, we're saying what happens when you actually change that letter. So what you're doing here in a missense mutation, you're changing the amino acid you're coding for. Okay, so what we mean by missense is, if we look at this table, so this is a codon one, because we can tell it's RNA because we've got our U's in here rather than having T's. So say I had something that was originally CUU, okay, and I changed the middle letter here. So instead of it saying CUU, I now said CCU. Now that would change it from this amino acid, leucine, to this amino acid, proline. Now, you need to know more about the amino acids to make a judgment about whether that's actually a really bad thing. It might be that what you've shifted it to is something that's got quite a similar feel. Like it behaves in the same way. It's got the same kind of chemistry. So shifting it is maybe not going to do a major deal. But at the same time, you might shift it to something that is completely different in the terms of its chemistry, changes the fold of the protein, means it can't connect somewhere where it should, and it completely wrecks the protein. You just don't know. But the important thing is that we have got a miss, as in it's not right, sense. So sense is saying something. So we're still coding for an amino acid, but we're now coding for a different amino acid. So if we code for something which is very similar, okay, so our example here is lysine, arginine, and histidine are all basic. Okay, so here's histidine, where we're going. Lysine cannot find at the moment. Where's my lysine? Oh, this is annoying. There we go. Lysine and arginine. So arginine over here and arginine over here. So if I shifted, say I had lysine with AAA, if I changed this code here to G, I actually shift it into arginine. So although I've changed the amino acid, I have done a missense, I've not really shifted the chemical behavior. So hopefully that won't actually do a big change to the protein. And you could see how some of these things could work. I'd forgotten that I had animated that. I could have just clicked that button. So anyway, if you change the code and get an amino acid the same type, little impact. So therefore it's neutral. But you are still changing the amino acid, so it still makes sense. Worse would be if we had a nonsense mutation. So nonsense means that I now make absolutely no sense whatsoever. So I am not saying any amino acid word. So what I'm now saying is stop. Okay, there's no sense at all to it. So it gives you a shorter protein. So if I shift anything to these three, so say I have a code for tryptophan here with UGG, and I change this G to an A, I suddenly now have a stop. Now, if that's early on in the protein, the whole thing is going to be wrecked. If it's late on, that's probably still going to be wrecked. It's going to be a real problem for the protein. They might survive it if it's very close to the end, but generally it's likely to cause real problems. Just as an aside, silent mutations are also possible. So a mutation there doesn't change the amino acid code. So this is not a missense. What we've got here is because of the redundancy in the code. So here, if we look at all these ones kind of connecting up here, phenylalanine at the top here is UUU or UUC. That means if I actually changed my U to a C or a C to a U, it wouldn't matter. So you could be sitting next to someone who is producing exactly the same protein as you with exactly the same amino acids, but your code is different because there is redundancy in the code. And we would never know unless you did a sequence and actually looked to see exactly what was going on there. Okay, so silent mutations are possible. You could have a mutation and never know. Right, so here's your last one. If you hit a mutation event on a splice site that would stop you doing a normal splice, so it's where you recognize that that's an intron and that's an exon, then hopefully you can see that this is going to completely mess up the whole thing. So if I say that I had, say I've got a series of exons here, and let's put a little series of introns in between. So here we've got some introns that we would normally just cut out by recognizing the sites. So you've got this is a site here, and you know that each of these have to be cut. Right? If a mutation event comes in and gets rid of a cut site, well then you wouldn't recognize it has to happen. So the first place you cut is here. So you leave this intron in, and you could do that in various ways. You could either lose exons or you leave in introns, and either of those is not good, and you're going to end up with a protein which is likely non-functional. And then we have our last type. Okay, so we've got substitution could do any of those things. Insertion and deletion we can look at one type, because what they are frame shift mutations. So what we mean by frame shift is we're looking at the reading frame of the gene. So deletion, I'm hoping we would see automatically that has to be delete a base. Okay, so this is what I mean by reading frame. Here is reading frame 1. So what we're saying is I am starting reading at this point here, and then a reading frame is a triplet because that's what we have as a codon. So I read, that's my first one, there's my second, there's my third, and that's what I'm doing. But there's a second reading frame which instead of starting there, I could start here. So that means actually in this one, this is my codon, and there's my next one, and there's my next one, and I could just keep on going along going, these are my codons for this reading frame, this is reading frame 2. And then finally, we've got a third reading frame, which is obviously just one further along. Okay, so let's assume that we are working normally in reading frame 1. And then we delete a base. So my sequence changes. Now I start with my normal reading frame, that's fine. I go ATT and I'm here. And then I go to my next one and that's still okay. But now I'm actually, because I've deleted off one of the bases, I actually end up shifting my reading frame. So I am here and I'm going to this one here to get my triplet. So now I've shifted into reading frame 2. And now all of my code after this is now in reading frame 2. And it's all rect. This is all different. Every single amino acid downstream of where we actually hit that deletion has been changed. And exactly the same thing happens if I do insertion, except this time we will go to the other reading frame. Okay, here's 1, here's 2, here's 3 again. Right, insert an A here. We've now got a new sequence. Okay, first one reads okay, next one is still okay, next one is okay, and the next one shifts us into reading frame 3. And now everything down here is all in the wrong reading frame. So we've completely changed the protein all the way after it. It is likely to completely wreck it. Okay, so your most drastic forms of mutation are probably insertion, deletion, and nonsense. Okay, that's the first part of this.